So good morning, dear colleagues. Pleasure to see all of you again. And thank you both, Don and Marcello, for the introduction. Maybe a little bit of history. So, you know, I suppose with you it's the same. Somewhere in your research career, you read a book or you read a paper that changes your life. I've had 3 of those. 1979, I read the Catcher and Burns paper on the control of flux, and that led me into how metabolism is controlled and regulated. 1995, I read Robert Rosen's Life Itself, and that completely played with my mind and changed the way I looked at biology. And then in 2004, I read the Organic Codes, which also changed my whole way of looking at biology. And I had the great pleasure of having Marcello and Joachim de Boele with me in Stellenbosch at the Stellenbosch Institute for Advanced Study, where we actually planned the International Society for Code Biology together, put together a bibliography, put together a glossary. And Marcello became my very, very dear friend. And it's for me also a great pleasure to pay homage to you today for years of friendship and inspiration. So, let's talk about a real code and not just superficially about how fantastic codes answer. So, I must first just make a disclaimer. What I'm talking about is not my field of expertise. I'm not a quorum-sensing expert, but it's such an interesting code that I feel at some stage somebody should say something about it, and perhaps I can inspire someone to write the paper. I'm not going to do it, but maybe someone else can do it, because the code is not in the list of 237 because there's nothing been published about the code. I mean, lots have been published about quorum sensing, but not the fact that it is a code. And that's what I want to do. So this is one of the first slides that I think it was at the Paris meeting that I had the slide, and it also sums up what Don showed just now. So Marcello makes the point. In the beginning, there was a genetic code, and then 3.8 billion years later, cultural codes. And the question is, nothing in between? That was his original question, which is a very good one. And of course, there are lots of them. We know now 237 published ones, which led to code biology. However, very early in evolution, bacteria discovered a whole set of new codes that allowed them to talk to each other within species, between species, and suddenly we have a group behavior. So I grew up with the idea in the 70s when I was a student, bacteria are free living organisms and they got nothing to do with each other. They just, carry on and by dividing and eating stuff and getting more and dying and whatever. Now we know it's completely different. There's a whole set of community codes. Like we have cultural codes, they have a sort of a primitive cultural code. And that's what I want to talk about today, because I find it absolutely fascinating. So what is quorum sensing? The ability to detect and respond to cell population density. This is the key word. By physiological responses through gene regulation. Those are the concepts that are involved in quorum sensing. It was discovered in 1970, that's how many years ago? 50 years ago, 53 years ago, by Nielsen, Platt, and Hastings. In one particular organism, the bioluminescent marine bacterium Vibrio fischeri, also known as Alii Vibrio fischeri. So what they found is when they grew these bacteria in the lab, they did not luminesce, give light, in freshly inoculated cultures, but only after the bacterial population density had increased significantly. So if you put that flask and you sit in an incubated room which is dark, and you let it go, you can see nothing. But if you sit long enough, suddenly, in an instant, the light goes on. So all those bacteria at the same time change their physiological response and start bioluminesce. So because they attributed the conditioning of the medium to the growing population of cells, they refer to this phenomenon as autoinduction. And so I want to explain to you today how this autoinduction works. So it was also discovered after that in another Vibrio harveyi, which also bioluminesces in the same way. The difference between the two is that Vibrio fischeri is a symbiont. Vibrio harveyi is a free-living marine bacterium that also is pathogenic. But I have to tell you the story, perhaps some of you have heard this, about the symbiosis. And it's the symbiosis between Vibrio fischeri and a little squid, that size, half of my thumb, called the Hawaiian bobtail squid, Euprimus colopis. Now, you must imagine this squid lives in the shallow waters around Hawaii, and it has a problem. The problem is that it's a predator at night, but the moon shines from the top, and because of the shallowness of the water, it casts a shadow, and the shadow alerts its prey. So how does it compensate for the shadow? Well, sorry. Let me see if I can. How did we manage to get to get this thing full screen?
Just click on that.
Just show. Which one? That one.
Yes.
There we go. It has in its mantle, it has sacs, in its ink sacs, a place where the Vibrio fischeri can live. And it actually feeds them. It's a symbiotic relationship. It provides all the nutrients. And in those two sacs there, as shown here, and you can see these ones here, the Vibrio fischeri grow. And during the day they grow to the point in the night where they are enough that they can give light. What it has in these sacs here, it has a membrane that can shift. So what it does when it swims, it has a sensor on the back that senses what the moonlight is, and at the bottom it counter-illuminates. So it compensates for the light that comes in so that it doesn't cast a shadow. Right? It's absolutely fantastic. So it uses the light of the bacteria to make itself, it's like Harry Potter, the invisible cloak, yeah, invisibility cloak, exactly the same. But of course, the bacteria keep on growing because it gets food. So at night, when it's now finished, what it does in the beginning of the day, it goes under the sand where it hides for the day, and then it pumps out most of the bacteria. And then it starts growing again. And by nighttime, the whole cell population has grown to a high enough density so that it provides light again. It's an absolutely fantastic story, so I love it. I have to tell it. So, in order to understand how this works, this autoinduction, let's first just have a look at chemical signaling in general. You usually have in cells from eukaryotic organisms up to mammals, 4 types: autocrine, direct, paracrine, endocrine. So autocrine is when a cell produces a signal that acts on itself. Direct signaling is when 2 cells are so closely connected through gap junction and so on that it's possible for one cell to communicate to the other one by molecules just diffusing through the gap junction. Paracrine means closely associated cells, giving a signal from one to the other, like, for instance, neurotransmitters in a synapse during the synaptic junction. And then endocrine is like hormones. My adrenal glands produce a steroid hormone that goes somewhere else and has some other effect. So you have various types of signaling molecules associated with each of those forms of chemical cell signaling. On the other hand, you have 2 types of signal transduction. So I'm going to talk a little bit just now about the basic mechanics of signal transduction, but here we have either an intracellular receptor. So the signaling molecule comes from the outside, let's call it the first messenger, but it doesn't produce a second messenger. It actually diffuses through the cell membrane, binds to an intracellular receptor. The typical type of signaling molecule is a steroid hormone, and then that complex has some other effect on genetic transcription. So it is like a transcription factor, which then promotes or represses the transcription of genes. On the other hand, you have the classical one, where you have a membrane receptor. The signaling molecule binds on the outside, and on the inside something happens. Either enzymatic production of some other second messenger, Marcello had in his slides, or you actually had in the slide, the typical ones: calcium, diacyl glycerol, inositol triphosphate, and cyclic AMP or cyclic GMP. Those are the typical second messengers. But what can also happen is that the receptor, when it binds on the outside, it activates an enzyme on the inside that starts a phosphorylation cascade that leads to some other gene effect on transcription. So it's either via intracellular receptor, via a membrane receptor. The reason why I show these different parts is because quorum sensing, in a sense, can use all of these. So it's at the same time an autocrine and an endocrine because it affects itself and it also affects the others, right? And it has two types of mechanisms, both of these intracellular and membrane receptor type of mechanisms. So bacterial quorum sensing uses all of these. How does it work? As an example, I'm only going to use the one where there's an intracellular receptor. So what happens, there's an enzymatic reaction that produces inside the cell a chemical called an autoinducer, which then diffuses out of the cell through the membrane, so that the intracellular concentration, extracellular concentration is always the same. But now remember, all the bacteria are doing this at the same time. So as the bacterial population increases, the concentration of these red dots or these red triangles on the outside, they also increase. Of course, they are inside and outside, so at a particular concentration, a threshold concentration, the molecule, the autoinducer, binds to a receptor inside, right? Like the steroid hormone would bind to a receptor on the inside, except that steroid hormones come from the outside. These guys are produced from the inside. That binds to some other promoter region on a gene, and it has some other physiological response through gene transcription. That's very simply how quorum sensing works. It can have a huge variety of physiological responses. I've listed a few: bioluminescence, symbiosis, virulence, competence, conjugation, antibiotic production, motility, sporulation, biofilm formation. In your gut, as you sit there now, all of those are happening at the same time in the very complex microbiome community in your gut. Communication between bacteria and themselves and between you and the bacteria at the same time, because we also have developed mechanisms for detecting these autoinducers. So there's a cross-control. In fact, I'll show phages can also talk, bacteriophages, viruses, also talk to each other, and they talk to the bacterium, and the bacterium talks back. So maybe, Marcello, it's not true that the prokaryotes at some stage just stop. They are still developing codes. As we sit here, we just don't know it. But I guarantee you those codes have not stopped. The community codes, they are still being developed because the environment is changing all the time and they have to fight for their survival all the time.
No, what I did say is that don't develop codes that increase their complexity.
Yes, right. Well, but the community increases its complexity. No. That's the interesting thing for me. Oops. So let us, I'm going to irritate a bit of, because we all know what the code is, but just let's recap this. So what is an organic code? It's the translation of an organic sign into an organic meaning via a set of conventional rules. But in order to be able to do that, there must be some other adapter between the two, like there is for the Morse code operator a piece of paper that says A is da, B is da, dit. I was in the Navy for a year. I was a Morse code operator. So for me, that is a very nice way of thinking about it. So what happens? The adapter molecule recognizes by binding the organic sign and becomes an identified organic sign, which is then translated into an organic medium. Let's just look at one example, the signal transduction code, because this is, of course, what autoinduction also is. So you have a first messenger. You have either a transmembrane protein or an intracellular receptor protein that recognizes the first messenger, either on the outside or after it's diffused inside, and it's then translated into a physiological response, which can either be a second messenger, like I mentioned just now, or it can be directly a gene transcription response. So in general, it can be just a physiological response. So what we have is, on the one hand, non-covalent binding of the organic sign, and then some other translation, either enzymatic or genetic, into some other organic meaning. And the two, connecting the two, is some other convention. And very important, it's not a chemical transformation. It is a non-deterministic coupling between organic sign and organic meaning. So how can we translate these ideas into the autoinduction code. So we have exactly the same. We have an organic sign being produced, the autoinducer. It diffuses out. It binds to the adapter inside, which is the receptor. That then binds to some other promoter on the genome, and it has a response. So this is a perfect example of a organic code. And then we have, of course, that is then what I've got circled here in light blue, is the organic meaning of the organic sign. So let's translate it into reality. How does Vibrio fischeri do it? So there's an enzyme called LUX1, which converts S-adinosyl methionine into an autoinducer called an acyl homoserine lactone, which I've shown at the top here. That is the autoinducer molecule. That then diffuses out until it reaches a certain concentration. Remember, all the bacteria are producing this. It then binds to a protein called LUXR inside the cell, and LUXR then binds to the promoter region of an operon. As you can see, there are 1, 2, 3, 4, 5, 6 genes in that operon. And it also, at the same time, promotes the LUXR itself, its own production. So what you have is when this effect kicks in, you have on the one hand, you have the genes for, that's the LUXI, sorry, that's the ABE part, which encodes the enzyme called luciferase, which then in this reaction causes the production of light, which is then the physiological response. But it also increases the concentration of LUXI itself and of LUXR. So there's a sort of a positive feedback effect inside the cell for this as well. So this is the mechanism by which Vibrio fischeri synthesizes the autoinducer and how the effect in the end comes to be. This is just the reaction by which the autoinducer is produced. For those biochemists like me, that's interesting, but for the rest, it's just, okay, that's produced. There have now been discovered a whole host of these autoinducers produced by different types and different species of bacteria. Usually the gram-negative bacteria produce these acyl homoserine lactone inducers, but you can see that they basically have the same structure. They all have this lactone here, but they have different side chains to it. Gram-positive bacteria usually have oligopeptide autoinducers. And you can see they also have different bacterial species, and they all have a different structure. And then it was found that many bacteria produce this particular autoinducer, called autoinducer 2. I'll show the structure again. And that is supposedly, according to some at least, a general one. That general, let's call it a universal signaling molecule for the bacteria, so that they don't only listen to themselves, but they also listen to other species at the same time. But it's not clear whether it is really universal or whether it only works within subgroups. So here are the two quorum sensing mechanisms. The one we've now discussed, which is where the autoinducer diffuses back and binds to an intracellular receptor, but you also have the classical type of signal transduction, where you have a protein, which is a membrane protein sitting here. Let's say this is a precursor peptide now for the gram-positive bacteria. Gets transported out, doesn't diffuse out. You have an ABC transporter that takes it out of the cell, and then it binds to the receptor, activates the receptor, which is a histidine kinase, and that sets into motion a phosphorylation cascade that leads in the end to a phosphorylated response regulator, which then binds to a phosphorylated response regulator, which binds to some promoter and activates or represses the transcription of some target genes. So you have both of these mechanisms operating at the same time. So just you can see here, this is just comparing for Vibrio harveyi, low cell density. You can see it leads to phosphorylation. High cell density, when the autoinducer binds to the intracellular receptors, you have an opposite effect of phosphatases that dephosphorylate. And in this case, you have some quorum regulatory RNA synthesized. In that case, it's not synthesized. Here you have no transcription of Lux R. There you have transcription of Lux R. So I've taken this from another slide so the colors don't match. But what happens here is that in this condition, you have the low cell density, you have virulence and biofilm production. But if you have high cell density, then it works the other way. Then these are suppressed, and you have the transcription of the activation of Lux R, which then leads to bioluminescence in the Harvey eye. Right. Very quickly now, because that's basically what the mechanism is. So the autoinducer, as I said, this one was discovered, and they thought that it was a universal signaling molecule. We're not quite sure, but it seems that they probably act exclusively between subsets of bacteria in mixed species communities. There was another one very recently discovered, a pyrazine called autoinducer 3, that also seems to be either universal or partially universal. Okay, just to show you that all of these bacteria have a similar type of autoinduction system, but they use different mechanisms. So I'm not going to discuss the mechanisms, but here, for instance, is one for Staphylococcus aureus, Pseudomonas aeruginosa, Bacillus subtilis. You can see they're all different, but the principle is the same. So, to summarize, at low cell density, you have the production of these autoinducers, various kinds, and at high cell density, they diffuse back into the cell or transported back into the cell, bind to their receptors, intracellular receptors, and that then leads to some other group behavior in the bacteria. But what we also have discovered, and that's very recent, is that bacteriophages also do the same type of thing. So here is a bacteriophage that produces from its own phage DNA, it's called the arbitrium phage DNA. It produces a precursor peptide, which diffuses out of the cell, gets activated by splitting off a part of the oligopeptide, and then you have the so-called mature arbitrium peptide. Now, if there's not enough of it outside, you have a whole sequence of effects which leads in the end to lysis of the bacteria. But in a high infection state, when you have a lot of these arbitrium peptides, it suppresses the lysis, and you go into a condition of lysogeny, which means that the RNA, the DNA of the virus gets incorporated in the DNA of the bacterium. We also know that there's a bacteriophage communication. So the host produces a compound called DPO, and what the vibriophage, in this case vibriophage VP8A2 does, it makes a receptor which is analogous to the host receptor. So it hijacks the host system, natural host system. In this case, it leads to lysogeny. If there's a lot of DPO on the outside, host-produced DPO, then it produces lysis. We know now there's also the other way, communication the other way, but this is very, very recent work. So, to summarize, bacteria can communicate within species or between subsets, maybe of species, during these autoinducers. In that way, they can regulate a wide variety of group behaviors. Phages can communicate with each other via quorum-sense arbitrium-like peptides, and that allows them to regulate the lysis-lysogeny cycles of the host cells. And then recent results suggest that some phages can detect the host quorum-sensing systems and vice versa, allowing them to control each other's behavior. And all of this is done through the quorum-sensing codes. Thank you very much.
You must write the paper on Quorum-Sensing Code. Only you can do it. Come on, Jan. It's something,
Can I finish my book first?
I think your paper is short.
I spoke to Robert. Maybe Robert and I can do something together, because Robert also recognized that there were quorum-sensing codes. He emailed me as well and said he's very interested, but he couldn't put it in his list of codes because there's no publication about it.
No,
I thought I could perhaps inspire somebody else here.
wait a minute, you are the only person who can write that thing. You must write it, Yanni. You don't want to write
enough.
Quorum-Sensing Code by Yanni HOFMEYR. That's it.
But isn't it fascinating? I was really bowled over. So if you want to know something about Quorum-Sensing Codes, there's a researcher called Bonnie Bassler, and she's got an incredibly nice TED Talk. You'll recognize when you listen to a TED Talk where I got some of my stories from. But if you look at the bottom where I quoted, most of this comes out of her and her lab's work. So they've done an enormous amount of work on quorum sensing. And she's a wonderful communicator.
Quorum-Sensing Code.
No.
Here's the point. Here's the point.
Did you check the quorum sensing of plants? Because you know the plants, the roots of the plants, check whether other plants of the same species or other species are around. They do different things depending on whether there are enemies or friends around.
So I'm not sure whether those are auto-inducers or just by like hormones. So
I don't know.
But it's worth. I know. It is worth checking. Absolutely. Yeah.
You say that they interact so that some of the entities control the others. Is it control the right word, or the interaction induces the other part to react?
Well, I would say if I kill you, have I controlled you? Yes, it's a form of control. I kill you, or I protect myself against that sort of effect. So maybe control is the right word to use. I'm not sure. Maybe can you suggest something else? Maybe
An entity is a non-linear reaction, so it is the opposite of interaction. So if you kill the entity, you will no longer be able to.
So maybe that's not a good example. But control means affecting behavior, and I think they can affect each other's behavior. And in that sense, it may be an appropriate term.
I'm an engineer. For me, control is a very deterministic way of conditioning the behavior of the other part.
Yeah, but I think actually, I think that is a very nice way of putting it. That's what they're doing to each other. They're conditioning each other's behavior. Yeah. But that's a good question. Huh? Well, always be careful with words.
And they're conditioning it through the environment, right? Because when this is coming from the outside, none of this happens.
Exactly.
They'll recognize
Yeah.
that coordinates the behavior.
Yeah.
So again, I think you can take these ideas, and again, it applies to microbiome, it applies to ecosystems. Very, very timely discussion. I certainly second and third, fourth the idea that you should write this up, because really it applies to so many high-level issues, both inside of us and outside of us. What we do into the environment and all of this stuff, upstream and downstream cascading effects. I think it's fantastic.
Also for pathogenity. I mean, so if you get infected with Staph aureus, a horrible one in hospitals, it hides. You don't know about it. So it hides in your body, and it grows and grows and grows until it reaches that cell population density that allows it to be effective when it switches on its pathogenicity. The horrible thing. So the interesting thing is also, can we learn to manipulate quorum sensing in order to protect ourselves against infections? That's a huge part of the research at the moment.
In a way, quorum sensing is the way in which bacteria form a population.
Yeah.
That's the idea of developing a multicellular organism. That's
exactly. That's the precursor. So you have this paracrine, and at some stage they came together and formed a real entity, which you cannot divide anymore.
Yeah, that's what I mean.
So biofilms, I suppose, was the place where really eukaryotic organisms started existing within a biofilm, where they're very closely associated with each other.
I wanted to add what my fellow said. There are another set of codes, what I would call the biofilm formation codes. It's highly related to this density topic. And a colleague of mine who found a gene mutation that in the cell, when a certain density is in this bacterium, it forms biofilms. And so it's density dependent, but it's a somewhat different topic, the biofilms, compared to all the other quorum sensing effects. But it's, on the same hand, it's related to this.
Yeah, but the biofilm formation, as far as I understand it, is the effect of a quorum sensing code. I had it also with HOFMEYR, certainly. HOFMEYR has biofilm formation either on or off because of its quorum sensing. So maybe the two are very closely related and not necessarily separate. But I don't know.
Last one?
I'm also thinking that might be a good ground for conciliation with other views of how those go. And I'm particularly thinking of analogical digital transitions, because it seems to me that all those are ways of making something which is stochastic, deterministic at a certain point, and creating state transitions, like in bacteria between aggregating or adhere to
Yeah.
the substrate.
Yeah.
And then it becomes a digital choice, because codes are coupled.
Yes, if your response is going to be on-off type of thing, then one can regard it as a sort of a digital code, maybe. So I don't know. Yeah,
And
yeah,
do it without interpretation.
yeah. It just happens. Yeah, it's just built in. Yeah, the response is built in. Yeah, thanks, Marcella.
Interesting,
Thank you.
beautiful.
Okay, thank you.
Well,
Thank you, Jan.
Right.
Thank you.
we have now 30 minutes of coffee break. Please enjoy.
All right.